Bismillahirrahmanirrahim. Assalamu alaikum. Dear students, I welcome you in Pakistan International School Types virtual learning class of biology for grade first year. Dear students, today's lecture number 21, and we are on chapter number 2, that is biological molecules. Dear students, in the previous lecture, we have studied about nucleic acids. So this is the sequence of that topic, because without concept of gene, there is this topic cannot be completed. So let's talk about today about genes, as well as we will talk about RNA. So let's get started. Yes, students, if we see that genes, like what are genes? So genes are the sequence of nucleotides, because these nucleotides are the part of DNA. So genes are present on DNA. So we can say that gene is the physical and functional unit of heredity. Like genes transfers the information which the DNA contains from the parents to the next generation, and the information is taken to the next generation in the form of specific proteins. We know that our body depends on proteins for different tasks. Enzymes are proteins, hormones are proteins, antibodies are proteins, and muscles are proteins. So lots of functions which are carried out in our body are held by the proteins. So proteins are having that information which is given to them by the genes, which are transferred from parents to the next generation. So it means that this is true, that physical and functional unit of heredity is genes. So what is genes? Actually, it is the region of DNA and which is made up of nucleotides. It can vary in the length, like there can be from 20 to 200, more than 200 types of nucleotides in one gene. It depends on the characteristics like eye and hair color, height, intelligence, etc. So what happened while genes are transferred to the next generation? So genes from DNA that is copied into messenger RNA, and then it moves out of the nucleus, and they form polypeptide chains, and uh, then form nucleus. And then polypeptide chain can be twisted into many folds to make its different structures of proteins, and then they just give all the information to that individual. So for gene transfer, ribonucleic acid is very, very important, because DNA gives messages to RNA to form or to produce proteins of required character. So to make protein, first of all, we have to study about RNA. So let's see this structure. At first, we will talk about a brief structure, with the diagram. You can see here, this is chromosome. Then this is chromosome is the condensed structure of DNA. So you can see there is double helical structure. We know that RNA is a single helical structure, like it is one part of that. The formation of RNA is done by the transcription of DNA, like DNA is double-stranded structure, it opens up and RNA make the copy of this DNA sequence, what is the sequence which is required for the specific character. These two helical like backbones, or these two helical this chain, these chains are just broken down from these hydrogen bonds by the enzyme, and by this breaking apart, these two chains are separating apart, and from one chain what happened, um, the DNA make another copy of that, that is the complementary of DNA. We know that in place of thymine there will be a uracil at that RNA. And now this RNA which is formed here, that is called messenger RNA. Like it takes the messages of that part of the DNA, or the part of the that gene which the character has to be transferred to the next cell or next generation. If we see, this is RNA which is transcribed from this DNA after breaking this one. Like if it is A, so it means if this one or this one, any type of can be this chain, this polymer chain of this DNA can be um, can have the information which is to be transferred to the RNA, and then the protein, required protein is to be formed. You can see here the process of transcription. This is DNA. Now it's breaking from here with the help of enzymes, and it makes the copy of its message which is given here. So that is the single-stranded helical structure. It's also having sugar phosphate backbone outward, and also all the we can say all the nitrogenous bases which are attached with that. In place of thymine, it is having uracil. So you can see here complete structure of this one. This is RNA, and because it is too long, so sometimes it is twisted around and make the double helical structure with the hydrogen bonding which is in between them. So you can see another process, like there is the replication of this DNA, and when it is required there can be transcription, means it gives messages to the RNA, and RNA makes a copy of this DNA, single copy, and then it goes to form the protein, that is called translation process. So from RNA to the protein, that is called translation. So that is actually protein synthesis process in which RNA is involved. So first of all, the process by which the message is transcribed in the form of RNA, that is called transcription. Then RNA changes into protein, that is called a translation process. Okay, so let's get started about RNA. RNA actually the polymer of nucleotide we have seen here. So it is single helical structure, as well as it is having in place of H group which is present at carbon number 2 in deoxyribonucleic acid, it is having OH group in carbon number 2 in ribonucleic acid. Okay, so RNA shows us RNA shows a secondary double-stranded structure in somewhere, as I told you in the last diagram. It is having three major classes or three major types. So these are called messenger RNA, ribosomal RNA, and transfer RNA. They are named as mRNA, rRNA, and tRNA. So what is messenger RNA? Messenger RNA is this as the same, not this one actually. Yes, this is messenger RNA which is transcribed from DNA, and it is its length depends upon the size of the gene as well as the protein for which it is taking a message. So, for example, a protein molecule which is having almost a protein molecule which is formed, it is having almost 100 amino acids. So, messenger RNA will have a 300 nucleotides in that, because for this every 3 nucleotides which are present here, they form codon, they form triplet, and they give like they carry messages in the form of triplets. So for a like for the one triplet or 3 nucleotides, there is one amino acid. So the transfer RNA which will transfer the amino acid to this one, it is having anticodon over it. This is codon, it means it is having the message. Okay, so this is in the form of triplet. For example, here can be C, G, and U, means uracil in place of thymine. So the anticodon will have G, C, and A. So the anticodon will be complementary of that with which the transfer RNA carries. So that codon, and that actually, so actually in the cell we are having the least amount of that, like 3 to 4% of total RNA in the cell. So when there is requirement of protein synthesis, this messenger RNA is transcribed from DNA, and it is carrying triplets of codons which are attached with the transfer RNA. So what happened to this messenger RNA? It will go out of the nucleus. For example, here messenger RNA is transcribed from DNA and it will move out of the nucleus. When it will move out of the nucleus, so you can see already here is a ribosome present, so it will attach on the ribosome. So here is, you can see the 1, 2, and 3 triplet of this one, UCG, and complementary of this codon is anticodon, which is present on transfer RNA AGC, which are attached with one another, and a transfer RNA also having one amino acid with that one, and the amino acid is attached with them. Then this amino acid, this transfer RNA will remove from here, and next transfer RNA will attach here in the next codon of this one. So messenger RNA will also move in this direction outward, and like that every amino acid will attach with and make the polypeptide chain for protein, for one protein. Almost 20 different amino acids will make one protein. There are 20 different amino acids. Okay. So this process is called translation, and that is for protein synthesis. Let's talk about ribosomal RNA. Actually, ribosomal RNA is composed of RNA and proteins. If you can see in this diagram, RNA and proteins are present here, and these are the sites for the attachment of transfer RNA, but we know that this ribosomal RNA, it is formed in the nucleolus, which is present in the a nucleus, and then it assembles out of the nucleus in the cytoplasm. And there is the large quantity of ribosomal RNA is present in the nucleus, sorry, in the cell. That is almost 80% of total RNA content is um, mm, RNA. The great number of this shows that 80% of that, because every time we need to make protein, so the ribosomal RNA must be present in the cytoplasm for protein synthesis. So they are present in two subunits, 40S and 60S, also larger and smaller subunits, and these two subunits, and together with some proteins, they are attached with one another, and messenger RNA attached here at this place. We know that transfer RNA is attached here at larger subunit, and messenger RNA will attach at the smaller subunits, and where the translation process takes place and protein is synthesized. Actually, this is the long chain of protein which just go into conformational change and form this very convoluted zigzag structure of a protein. And later on, after protein synthesis, these two subunits disassemble. Okay, the next one is very, very important as well. That is a transfer RNA. Transfer RNA means it transfers what? It transfers the amino acids into the protein chain. So actually this is the RNA. This is also transcribed from DNA, and it is the like we can say the smallest of all RNA, and it consists of just 75 to 90%, 90 nucleotides are there. It is also single-stranded, but it makes its structure somewhere duplex, as well as it is the structure like a clove-like. If you can see here, it's a transfer RNA structure. It appears a clove-like structure, and it is having a 3 loops. If you can see, the first the codon or the sequence of that at 3 prime, and that is always ACC, and the 5 prime and ends at G nucleotide. And actually this bound this like this is the end of this nucleotides, and nothing can bind here. ACC is the site where amino acid will bind. So this clove-like structure of transfer RNA is having 3 loops, and these loops are called D loop and theta loop, and in between it is having the attachment for for codon on the messenger RNA. So D loop recognizes the activation enzyme which activates the process of synthesis of protein, and a theta loop recognizes the specific place on the ribosome for binding during protein synthesis. Like at the back, if just imagine we are having a larger subunit of ribosome, and or like ribosomal RNA makes the ribosome, and here the smaller unit of ribosome. So this transfer RNA where it has to bind, because ribosome is also having 3 regions. Here it is attached, then it moves forward, and then it leaves the ribosome outside. So this theta loop just recognizes the specific place for binding on a ribosome. So after binding this, what happened? It is having anticodon, which is complementary to the codon, and it is having also the first amino acid of every protein, that is leucine. It attaches with that one, and there are almost 45 different kinds of transfer RNA are present in human beings, and 60 almost transfer RNA yet they are identified. So every amino acid carry different transfer RNA carries different amino acids. So there are almost 20 amino acids found in the proteins. So this helps in the translation of we can say translation of the proteins, like the message is completely translated into the proteins. Let's talk about the last topic of this chapter, that is conjugated molecules. Conjugated molecules, it means they are joined with other kinds of molecules, like different kinds of molecules are joined together. We have discussed them in lipids as well as in carbohydrates. So let's talk about once again, for example, glycolipids. It means carbohydrates is attached with the lipids, glycoproteins. Carbohydrate is attached with proteins, lipoproteins. Lipids are attached with proteins, and nucleoproteins, nucleic acids are attached with proteins. So first of all, we'll talk about glycolipids. They are complex lipids because they are combination of carbohydrates as well. So one or more simple sugars can be attached with that one, with long fatty acids and alcohol chains. We know about the lipids, and carbohydrates form a polar head of that molecule. So glycolipids are present in white matter of brain and myelin sheath of nerve fibers, as well as chloroplast membrane. Mm, Chloroplast membrane like this helps in the photosynthesis, as well as in the myelin sheath of nerve fibers, like then if this is the structure of a nerve. So this is the terminal and terminals of a nerve. This is cell body. So you can see this axon that is covered by a myelin sheath, or these Schwann cells is also called this a myelin sheath, and that is fatty in nature, like because it is made up of glycolipids, as well as it is found in the white matter of the brain. Brain is composed of white matter as well as gray matter. So white matter means it is white because there is glycolipids present, which is whitish in nature. So let's talk about glycoprotein. So that is formed by proteins and covalently attached carbohydrates. They are widely distributed in our cells because they are having the function as hormones are, like they can behave like glycoproteins, transport proteins or carrier proteins which carry the substances in and out of the cells, as well as structural proteins which are the parts of the cell membranes as well as the membranes of different organelles, as well as receptors. For example, this is the cell. On cell, there are receptors present. These are also made up of glycoproteins, which accept some sort of neurotransmitters or chemicals, and by that the gates open, and the conformational changes are done in the cell. Okay, so let's talk about lipoprotein. Means it attached a combination of protein and phospholipids are there. So phospholipid protein complexes are widely distributed in plants and animals materials. Like in the milk, we are having lipoprotein. Uh, yes, in the blood cell and nucleus as well, egg yolk or yes, egg yolk, it is also having like albumin membrane because that is rich in energy as well, and chloroplast of the plants. Let's talk about nucleoprotein, like consists of simple basic proteins with nucleic acid, and they are found in chromosomes and ribosomes. Uh, like nucleic acid, like DNA, is twisted around the histone protein. We know, we have studied the structure in grade 10, so that is also nucleoprotein. So Dear students, with this topic of conjugated molecules, Alhamdulillah, we have finished our chapter number 2. That was too much longer and very much conceptual chapter it was, and very helpful for the next coming chapters. So, inshallah, we will meet in the next lecture. Wish you all the best. Till then, Allah Hafiz.